Hey everyone, welcome back to Window Seat Studies. I hope you all are doing well. So today's topic of discussion is the dynamic changes in DNA methylation that happens during the development of an individual. So this video is going to be short and concise, discussing only this topic. I was supposed to post this topic after the DNA modification series, but anyways, let's start with the discussion. So today we are going to discuss about changes in DNA methylation that happens during the development of an individual. So we are going to discuss the important events that are going to happen from the fertilization of an organism to all the way to the formation of gametes. So all of us know that the life originates from a single cell known as zygote. So zygote is formed after fertilization of sperm and an egg. So sperm and egg, they fuse together and they form zygote. So zygote has its genetic material inherited from mother and father. So the material which is the genetic material which is inherited from mother is known as maternal genome, while the one which is inherited from father is known as paternal genome. So now this genome is inherited from parents, and that is why it will have its own distinct DNA methylation patterns. So it is going to have a distinct DNA methylation pattern. So that is why the very first event that zygote undergoes after the fertilization would be global DNA demethylation. Why? Because the embryo needs to set its own identity, and that will be set by the demethylation. So this demethylation is important because the embryo is going to set its own new methylation marks. So first, the whole genome. This event is global, means the whole genome undergoes the demethylation, except the imprinted genes. So it's like those are very important blueprints that gets reserved by the embryo. So except those imprinted genes, all of the methylation marks are reset. And for resetting, first event that zygote has to undergo is global demethylation. So the demethylation occurs throughout the genome except the imprinted genes. So this is an important point. After that, the embryo resets its genetic program, and that is why the second event that is going to happen is the DNA methylation. So this is the re-establishment of DNA methylation. Okay, so now the new methylation marks are they are set on the DNA of the embryo. The third event will happen. This will be the second demethylation event. The first was global demethylation event, but this second wave of demethylation, this happens only in primordial germ cells. So this is second wave of DNA demethylation. This only happens in primordial germ cells. Note it because it's very important. The first one is global except for imprinted genes, but the second wave happens only in the primordial germ cells. So those are the precursor cells that are going to form the gametes in the future, depending on the sex of the embryo. So this demethylation event is very important because after that there will be a sex specific establishment of the methylation mark in those primordial germ cells. Okay, so like for example, the embryo that is formed is going to be male in future. So it is going to produce sperms, and that sperms will have its share of the paternal genome in the future progeny. So that sperm is going to contribute in the paternal genome of the daughter progeny, and that is why the methylation patterns need to be set according to that. And this so for maintaining, so for generating that new methylation marks, the second wave of demethylation is done in primordial germ cell, and that after that, the sex-specific establishment of methylation marks is seen in those cells. So those are the main events that an embryo undergoes during the development. So let's plot this on a graph. So this is the y-axis and this is the x-axis. So the y-axis denotes percentage of DNA methylation. The highest represents 100%, means the whole genome is, so the methylation is 100%, means it's highly methylated, or it's not methylated, means it has undergone the demethylation. Okay, and on the x-axis represents time, means it represents the life stages, the stages of the life cycle. Okay, so the graph starts from here because before fertilization, the sperm and egg, both of them had their own methylation mark, and that is why the methylation percentage of DNA methylation will be high, because they already had their own methylation marks. So the first event happened is the global demethylation, and that is why the methylation marks will decrease and it will reach to zero. After it reaches to zero, there will be re-establishment of the methylation marks. Okay, and they will again reach to a high point, and now those methylation marks will be only demethylated. The DNA will be only demethylated in primordial germ cells. Okay, but then again the methylation marks will be re-established. Okay, so this is how the graph will look. But this, here comes the twist. This is only for the genome which is inherited from father. So this is for the paternal genome. What happens with the maternal genome? So the maternal genome graph starts from here. It undergoes the demethylation. Okay. Then again the methylation part, it's the same. And when it undergoes the remethylation in primordial germ cell, it will be low. So why is this? Why is this so? So what happens is that the sperms, this is the sperm, means the genome which is inherited from parent, and this is seen in egg. So this is in mother, the maternal genome, sorry. So this is maternal genome. So the sperm has less nucleus, and the whole genetic so the sperm has less cytoplasm, and all of the genetic material is very densely packed in the nucleus of the sperm. And for this dense packing, the sperm needs high DNA methylation compared to the maternal genome, and that is why the methylation would be high in the paternal genome compared to the maternal genome. Okay, so that is why the graph looks like this. And again, this re-establishment of the methylation mark is done during the either maturation of sperm or the maturation of egg. Okay, so this is after birth. This happens in primordial germ cells, the future gametes. Okay, so this is the global demethylation event. Okay, this line goes down to 0%. So the DNA demethylation has been done. This is the blastocyst stage of the development. Okay, and during the epiblast stage, the new methylation marks are made. Okay, so this is the condition of somatic cells. Okay, all of the somatic cells have high methylation. The methylation marks are re-established, and this is shown in the primordial germ cells. So this is how in the primordial germ cells, methylation marks are erased. They undergo the demethylation and re-establishment of methylation marks. So it will be high if the gametes are sperm, it will be low if the gametes are egg. The reason being this, that the sperm has the dense chromatin, they need the dense chromatin because of the high packaging in the nucleus. Okay, so the nucleus is very congested and you need the higher order of the packaging, and that is why it is done by the DNA methylation. So you know that DNA methylation supports the closed chromatin condition, and it is also associated to the repression of the genes. So this is the reason. Again, we are going to learn about some proteins known as PGC7 or Stella. Okay, so this is maternal protein. This is This maternal protein is this has a distinct function that it protects the genome from DNA demethylation. So we will discuss about the PGC7 and Stella in a different lecture. So that's it for today. These are the important events that happen during the development of an individual. So this is after birth, by the way. So this is after birth. Okay, and this is during this is the fertilization part. Okay. Okay, so during fertilization, both the paternal genome, the blue one represents paternal genome, and the pink one represents the maternal genome. So both of the genome, they have their own distinct methylation marks, and then they will be erased. It will go to the zero, and then it will increase the reestablishment of methylation marks in the somatic cells, and then some cells will be put aside, which are going to be constituted in the primordial germ cells because they are going to form gametes, and that is why the methylation there is DNA demethylation. Okay, this is DNA demethylation. It represents that the new methylation marks that will be set in the primordial germ cells will be according to the sex of the parent. Okay. So this is all about the DNA demethylation events, but this happens in normal, the somatic cells. In the imprinted regions, the imprinted genes, they stay as it is. Okay, so the imprinted genes, this is the imprinted genes. They do not get demethylated. The methylation marks are not erased in the imprinted genes. So that's it for today. In the next lecture, we will learn about genomic imprinting, and we will also learn about, yeah, we are yet to cover the X inactivation part. So first we will cover the X inactivation, X chromosome inactivation, which does the dosage compensation in mammals. The epigenetic regulation of X chromosome inactivation, the exist RNA, the T6 RNA. We will discuss all of this, and after that, we will discuss about genomic imprinting. Till then, see you. Bye bye.